Hi there, in this video we are going to discuss about the cell signaling. So what is cell signaling? Cell signaling is basically a methodology, or you can say mechanism, by how cells communicate with others. So the study of cell communication is the cell signaling, and this communication can be with other cells. It can be with extracellular matrix protein, and it can be with the extracellular environment. Okay, so this cell signaling is shown by both single or unicellular organisms, single cellular organisms, as well as multicellular organisms. Okay, single cellular organisms which involve bacteria. In bacteria, the cell signaling is necessary to grow, or you can say to obtain growth signals. It's necessary for them to search for their prey. Okay, and it's also necessary to obtain or search for their food, and to prevent themselves from toxic environmental agents, prevention from toxic agents, and so on. In multicellular organisms, there are billions and trillions of the cells. So in multicellular organisms, this cell signaling is necessary for the division of labor among the cells, and integration of the actions of various cells. So the cell signaling plays a crucial role in both unicellular organisms as well as in the multicellular organisms. Okay, before we proceed on in the cell signaling, we should be knowing about some general terminologies. In cell signaling, one term is ligand. One term is receptor. Okay, so receptor is basically a protein, or you can say a recipient which responds to some signal. So receptor is basically that structure, you can say a structure that senses physical or biochemical change. So the receptor is something which detects physical or biochemical change, while ligand is something or some chemical which binds to this receptor. Any chemical which binds to some receptor is called ligand. Okay, so this ligand, depending upon its you can say activity or action, this ligand can be, or you can say ligand can be agonist, or it can be antagonist. Okay, agonist is a ligand which increases the activity or response of the receptor, while antagonist is some ligand which decreases the activity of receptor. So ligand is classified into agonist and antagonist. Agonist increases the activity and antagonist decreases the activity. These agonist and antagonist both can be either physiological or can be pharmaceutical. Okay, so if the agonist or antagonist is naturally occurring, it is called physiological agonist or physiological antagonist. If agonist or antagonist is synthetic, made in pharmacy or industry, it is called pharmaceutical agonist or antagonist. For example, we see the example of physiological agonist and antagonist. Supposedly, uh, insulin decreases blood glucose, and glucagon works opposite to it and increases blood glucose. So, insulin and glucagon are the antagonists, and these are physiological antagonists. Okay. Now we move toward pharmaceutical antagonist. So, in pharmaceutical, there are you will study in third year in pharma subject. There are beta agonists as well as beta blockers. Beta agonist increase the activity at beta receptor, beta adrenergic receptor, and beta blocker block the activity at beta adrenergic receptors. So these are basically in the pharma subject. You need not to know the details here. Okay, so basic idea here is for you about ligand and about receptors. Okay, uh, next we move on about the let's see about the receptors. So receptors can be on the cell membrane. This is a cell and this is the nucleus, and these receptors can be on the cell membrane in the form of membrane proteins. Okay, and these are usually uh, protein receptors or polypeptide hormone receptors or catecholamine receptors. The hormones which are usually proteins or polypeptides or catecholamines. Catecholamine means epinephrine or epinephrine. Their receptors are usually on the cell membrane. The receptor can be in the cytosolic receptors, which is true for cortisol receptors in cytosol, and these receptors can be nuclear receptors located inside the nucleus, which is true for steroids. So these receptors, depending upon their location, they can be on the plasma membrane, they can be in the cytosol, and they can be inside the nucleus of the cell. On plasma membrane, the receptors are of protein hormone, polypeptides, and catecholamines, and on the cytosol, the receptors are of cortisol, and inside the nucleus, the receptor located are of steroids or retinoids and so on. Okay, so we have a basic idea of receptors here. Next, we should be having a basic idea of the messengers. Messengers are basically chemical substances through which chemical substances that lead to the cell communication. So chemical substances through which cell communicate are called chemical messengers. Okay, so these chemical messengers in the endocrine system, uh, they are called hormones. In immune system, these chemicals are called cytokines, and in central nervous system they are called neurotransmitters. Okay, so depending upon their locations, location, we can also classify them. For example, they can be autocrine if chemical released by cell acts on itself. Autocrine that act on same cell or same population or same type of cells are called autocrine. For example, cytokines released by WBCs activate either same white blood cell or the same class of blood cells. So there are autocrine chemical messengers. Okay, so they can also be paracrine. What are paracrine? Paracrine are those messengers which act on neighboring cells. Neighboring cells. For example, we take an example of synapse here. Supposedly this is a synapse, this is a pre-synaptic membrane, and this is a post-synaptic membrane. Here neurotransmitter acetylcholine is released and it acts on the adjacent cell. Okay, so this is acting on the adjacent cells, and this next neuron may or may not be same, or it may be a muscle cell at the neuromuscular junction. So this is an example of paracrine transmission. So they can be autocrine, they can be paracrine, and they can be endocrine or endocrine hormones which are transferred by blood to the distant sites, for example insulin, glucagon, thyroid hormones, and so on. So it was a basic idea about receptors and chemical messengers. So next we move on. Next we move on about a general idea of signaling, how signaling occurs. So in general idea of signaling, what happens, one cell is stimulated, there is a stimulus which activates one particular cell or gland. This cell or gland releases some chemical messenger. This chemical messenger goes to the target cell. In target cell it binds to the receptors. Then there is conformational change in the receptor, and then the receptor leads to the response or effector response. And this effector can be either receptor itself, or receptor can communicate with some coupling reaction, or can lead to some coupling reaction in which mostly G proteins are involved. Okay, so what happens, stimulus activates some cell or gland which releases a chemical messenger. This chemical messenger travels to the target cells, and the target cell it binds to the receptor, and this receptor lead to the effector's response, and this effector can be either receptor itself, or it can lead to the coupling with some other protein. Coupling means you can say receptor communicates with some other protein, and this protein in turn communicates to the effector. So receptor response occurs by either enzymatic activity of receptor itself, or receptor communicates with some coupling protein which is usually G protein to activate some enzyme to lead to its action. So it is a basic overview of the signaling. So next we move on about some principal mechanisms of the signaling. Then we will be understanding. Before moving to the principal mechanism, we should first discuss some communication mechanism of the cell. How cell can communicate? So cell basically communicate by 3-4 ways. One is direct communication, which is dependent upon the cellular contact, and other is without contact. Other communication is without contact. So this contact based occurs by gap junctions. Cells in the tight tissues are connected to each other by gap junctions. Positively, this is a cell here, this is a cell, this is a nucleus, and this is also a cell, this is nucleus. These cells are connected to each other by gap junctions. They will be also connected to it by gap junctions, and so on. These gap junctions are made up of connexin protein, and through these gap junction materials flow between the cells, and they are in direct communication. So this is one kind of signaling which is by direct contact of the cells. Another kind of signaling occurs by cell surface proteins. Okay, this is positively a cell here, and it has a protein here, and this is another cell, it also has a protein here, and these proteins interact, through which signaling occurs and reactions are regulated. So it is commonly shown in WBCs. For example, leukocyte have fast ligand, which is a, you can call it a protein name, a receptor name, and some target cell have fast receptor. Okay, so what happens when a leukocyte having fast ligand recognizes some cell with fast receptor and this binding occurs, it leads to the apoptosis of the cells. Apoptosis, which you will study in third year, the mechanism of apoptosis, in which that is written in detail how fast receptor and fast ligand interact and lead to the apoptosis of the cell. So this is why this is a communication which is also cells are having contact with each other, and it is occurring by cell surface proteins. Cell surface proteins. Next is without contact, and this without contact is by secondary messenger system, in which one cell releases some hormone. This hormone reaches to other cells, bind to its receptor, and triggers the intercellular response. So this is without contact. Moreover, cells are also in contact with the extracellular matrix, and they also respond to the molecular structures present in the extracellular space, and in response their cytoskeletal changes, and this is commonly manifested in leukocyte migration to the target site, which is also written in detail in third year in the pathology subject of third year. So it is basically how cells communicate with each other. So there are four mechanisms. One is by gap junction, cell surface proteins, by hormones or secondary messengers, and by their interaction with the extracellular matrix. Next we move on about the principal mechanism of the cell signaling. So principal mechanism of cell signaling are basically 6 here. One are gated. First one is gated ion channels. Second one is receptor proteins. Third one is receptor, and second one is receptor enzymes, and third one is receptor proteins. Okay, fourth are nuclear receptors. Fifth are receptors without enzymatic activity. Sixth are adhesion receptors. Okay, so gated ion channels are basically ion channels which respond to the stimuli, and they are basically ion channels which respond to the stimuli, and these ion channels can be voltage gated, or they can be ligand gated. Receptor enzymes are the receptors which directly have enzymatic activity, and their common example is insulin receptors. Receptor proteins don't directly have enzymatic activity, but they are coupled to the proteins, coupling proteins. These are usually G protein coupled receptors. Nuclear receptors are located inside the nucleus, or they interact with the nucleus of the cell, and these are usually steroid receptors. Receptors without enzymatic activity, they usually don't have itself enzymatic activity, but they lead to the enzymatic response. Okay, next one are adhesion receptors, which are basically involved in the extracellular matrix communication. So these are principally 6 type of receptor which lead to the different type of cell signaling. So we will study them one by one. So the first receptor we will study are gated ion channels. Gated ion channels, which we discussed that they are either voltage gated or they are ligand gated. First we see both of them at a time. So firstly we should know about sodium channel. Sodium channel which is present in the cell membrane. uh, Sodium channel basically in this way, if I am going to draw here, this is positively sodium channel. There is one gate and this is another gate, and just positively this is the cell membrane here. Okay, these are lipids and so on. So positively this is a sodium channel. This is outside of the cell and this is inside. So this sodium channel has two gates. One is activation gate which is outside the cell, and another is inactivation gate which is inside the cell. So normally what happens, normally this inactivation gate is opened when the cell membrane is usually negative inside. What happens, cell membrane has a sodium potassium pump which continues to throw 3 sodium outside and 2 potassium inside. As a result, inside of the cell becomes negative. So usually inside of the cell is negative, and when inside of the cell is negative, what happens, in activation gate is open. However, the activation gate outside the cell is closed. It is closed. So normally these sodium channels or voltage gated sodium channels are closed. So what happens if there is a stimulus in the form of some hormone or some physical stimulus? What happens, cell membrane become depolarized, and what it become depolarized transiently, the region or adjacent regions positive, this region becomes transiently active, it is become positive, and outside become negative. It stimulates these sodium channel. When this sodium channel becomes stimulated, what happens, its activation gate becomes open. And sodium rushes inside the cell, and when sodium rushes inside the cell, then this region becomes active and it becomes positive, and outside become negative. And this region then in turn stimulate the sodium channel located in the next region, or you can say sodium channel which will be located here will be activated, and in the same way its activation gate will open and sodium will rush inside and become positive, and so on. This series continue. However, when it become positive, or you can say potential inside continues moves towards positive, this inactivation gate starts closing and it closes, sodium channel become closed, and you can say then potassium channel located here, its gate opens and potassium diffuses out and membrane potential returns to normal. I guess it has gone complicated. Let me summarize here. uh, Normal cell membrane has negative inside is negative, outside is positive. So what happens when cell membrane is depolarized? Supposedly there is a stimulus here due to which inside became positive and outside became negative. Then the next region, sodium channel, will be activated. There are two gates of sodium channel. The outside gate will become active, open, and sodium will rush inside here and will depolarize this membrane segment. And when it will depolarize, its inactivation gate will close, and then potassium channel located here will open and potassium will diffuse out, and in the next millisecond its potential will return to the normal or negative. So in this way there will be propagation of the potential across the cell membrane. So this is how this sodium voltage gated sodium channel work. And next we discuss the ligand gated sodium channels. For ligand gated sodium channel, we come across a synaptic junction. Supposedly this is a presynaptic membrane and this is a postsynaptic membrane. So what happens, we will be covering ligand or voltage gated calcium channel also here. Supposedly membrane potential, this is membrane potential coming here, and this membrane potential reached at this presynaptic membrane, and what happens, it activates calcium channels. Voltage gated calcium channels become active, and these voltage gated calcium channels allow the calcium from outside to diffuse inside the cell. When this calcium comes inside, it binds with calmodulin protein, and calcium calmodulin complex is formed, and this complex activates calcium calmodulin dependent protein kinase. And this kinase, what it do, it phosphorylates synapsin protein. This synapsin protein is the protein which holds the vesicles containing acetylcholine with cytoskeleton. So when it becomes phosphorylated, it releases these vesicles, and these vesicles come here, they fuse to the membrane and release the acetylcholine into the extracellular space. So what happened here, membrane potential reached the presynaptic membrane, it activated calcium channel. In response to this membrane potential, these calcium channel become open. Calcium came inside, it bound to calmodulin protein, which is a protein present in the presynaptic membrane, and formed calcium calmodulin complex, which activated an enzyme kinase. Kinase is the enzyme which does phosphorylation, means it adds phosphate to the some protein. So these kinases become active, they phosphorylated synapsin protein, which is the protein holding these acetylcholine containing vesicles. When it became phosphorylated, it released them into the synaptic cleft. These acetylcholine molecules are released here. So what happens, on postsynaptic membrane there are acetylcholine receptors. These are acetylcholine receptors. So these acetylcholine receptors, what they do, they have 5 subunits, 2 alpha subunit, 1 beta subunit, 1 gamma subunit, 1 delta subunit, and there are 5 subunits, and these acetylcholine receptors bind to the 2 alpha subunits. When they bound to 2 alpha subunits, these acetylcholine receptors become active. So when these acetylcholine receptors become active, then this channel opens. This channel is basically, you can call it a ligand gated sodium channel. Okay, so when it bounds to acetylcholine, it becomes active and it allows sodium to diffuse inside. So when it allows sodium to diffuse inside, the postsynaptic membrane becomes positive inside and negative outside. So it becomes depolarized, and it also allows the nerve potential or action potential to propagate on the postsynaptic neuron. So in this way, voltage gated and ligand gated channels are communicating or working. So here, if I summarize, here were voltage gated sodium channels, and you can call it sodium, you can call it potassium, sodium, potassium, and so on. So when as action potential was moving here, firstly sodium channel was become active, sodium was moving in, then potassium was becoming active after a millisecond, and potassium was moving out. So this segment remained depolarized for a millisecond. Next this segment became depolarized. Next this segment became depolarized, and so on. So in this way, action potential reached the uh, presynaptic membrane, and in presynaptic membrane, voltage gated calcium channel become active. Calcium came inside, and it will lead to the release of acetylcholine. Then this acetylcholine bound to the ligand gated channel or ligand gated sodium channel, and sodium moves inside, and here the postsynaptic membrane became active. So it was a mechanism how the gated ion channel work. So we have discussed both voltage gated and ligand gated channels. Next we move on toward discussing the uh, about discussing the receptor enzymes. So principal example here is the insulin receptor here. So this is the cell membrane. Supposedly this is insulin receptor, it has a beta subunit and one is alpha subunit. Supposedly this is alpha subunit, this is beta subunit. Insulin comes and binds to the alpha subunit and induces the conformational change in the beta subunit as well. Inside segment of beta subunit acts as a tyrosine kinase enzyme. It means that it will phosphorylate the tyrosine residue of other proteins. So firstly, when insulin binds here, it autophosphorylates itself. Phosphate binds to itself, and in the next reaction, it phosphorylates insulin receptor substrate, which is a protein. It phosphorylates this protein. Insulin receptor substrate gets phosphorylated by this tyrosine kinase receptor located at this insulin receptor. So this insulin receptor substrate then lead to the intracellular actions, or you can say it leads to the downward functions, which lead to the uh, decrease in the plasma glucose level. All the function of insulin are then mediated through this insulin receptor substrate. So this is how receptor enzyme lead to the cell signaling.